Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 11th November 2020, and as all of you know, we are into the revision mode. We are revising today chapter number 8, cell structure and functions. This is very important, not only for the sake of CBSE, it's very important for the sake of UPSC NDA as well as NEET exams. Many MCQs will come from this chapter. Hence, have second time focus on this. And all living organisms, as all of you know, we observe around us, ranging from microscopic organisms such as say viroids to giant organisms, very big organisms like elephants, whales. These are all made up of cells. And some are composed of single cells, they are called unicellular organisms, and some are multicellular organisms having more than one cell. But whenever we study a form, structure and composition of cell, then we call it as cytology. So cytology is a part of biology. So what is a cell after all? The cells are smallest entity that are capable of independent existence and carrying out all essential functions of life. That's why we say it is a structural and functional unit of life. Cell is the structural and functional unit of life. When we say structure, it refers to size and shape. When we say function, the duty that it performs, the properties that it exhibits. In unicellular organisms like amoeba, paramecium, yeast, bacteria, single cell performs all the essential functions of life. There is no division of labor. Everything is to be done by that single cell only. Right from digestion, respiration, circulation, including reproduction, that single cell has to do. In the form of maybe, as all of you know, the fission, multiple fission, binary fission or spore formation. In multicellular organisms, different kinds of tissues perform different functions and have division of labor. Here, digestion will be done by digestive system, respiration will be done by respiratory system, like that. So there is division of labor in multicellular organisms. And we must be thankful to this gentleman, Anton van Leeuwenhoek, first saw and described a live cell. And of course, Robert Brown is credited with the discovery of the nucleus of the cell. And two scientists, we must be thankful to them for providing, that's Sweden and Swan, or Sleeden and Swan. In 1938 proposed cell theory that was of course modified by Rudolf Virchow. And all these scientist names you need to remember because of your UPSC exam. And all living organisms are composed of cells and products of cells. And all cells arise from pre-existing cells. So basically, two types of cells are there. One, prokaryotic, and second, eukaryotic. Pro means primitive, first formed. Karyon means nucleus, primitive nucleus. Cells that are having primitive nucleus means nucleus-like, we say nucleoid. And U means very clear, clarity is there. What is clarity? Karyon, nucleus of the cell. So cells that are having very clear and well-defined nucleus is called eukaryotic cells. So what is the difference between prokaryotic and eukaryotic? These are membrane-bound, nucleus is absent. There is no membrane, cellular membranes will not be there, organelles membranes will not be there, and nucleus itself is not very clear. Whereas in membrane-bound nucleus is present. And prokaryotic cells are small in size, cells are large in size in eukaryotic. Single chromosome is present, more than one chromosome is present, membrane-bound organelles are absent, membrane-bound organelles are present. These are the broad categories under which we differentiate. So shape and size of cell varies greatly according to their position and function. Mycoplasma is the smallest cell, UPSC, and largest isolated cell is ostrich egg of an ostrich. The shape of cell may be cuboid, columnar, polygonal, thread-like or irregular like in amoeba. So prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, PPLO, and they multiply rapidly and vary in size greatly. Bacterial cells may be rod-shaped, they are called bacillus. Spherical-shaped bacteria are called coccus. Comma-shaped ones are called vibrio, and spiral-shaped are called spirillum. Based on the shape, 4 types of bacteria. All prokaryotic cells have cell wall surrounding the cell membrane except in mycoplasma. Genetic material is naked. The plasmid DNA in some bacteria provides some special features like resistance to antibiotics. Of course, we talk about this, if you may recollect from biotechnology chapter. Plasmid, extra chromosomal circular DNA. So remember that, and it has got genes that resist to antibiotics. Cell organelles like mitochondria, Golgi bodies are absent in prokaryotes. A specialized differentiated cell membrane called mesosome is the characteristic of prokaryotes. And bacteria, based on the stain, whether they get colored or not, when color is applied, there are 2 types, gram positive and gram negative. So gram positive are they retain the stain, and gram negative, they don't take up the stain at all. So in bacterial cell, a chemical complex cell envelope is present which consists of 3 layers. The outermost is glycocalyx, middle one cell wall, and the innermost is the cell membrane. So there is an extra one, glycocalyx. Glycocalyx may be as loose sheath in some bacteria called slimy layer. In some other bacteria, glycocalyx may be thick and tough called capsule. Plasma membrane is semi-permeable membrane having mesosome in the form of vesicles, tubules and lamellae, long tubes. They help in cell wall formation, DNA replication and distribution to daughter cells. Motile bacterial cell, motile means movement, contain flagella, which is composed of a filament, hook and basal body. Remember that 9+2 structure. Pili and fimbriae are the other surface structures that help in bacteria to attach with host and other substances. In prokaryotes, ribosomes are attached with cell membrane having 2 subunits, 50S and 30S, to form together 70S prokaryotic ribosomes. S for Strasbourg. Ribosomes are site of protein synthesis. Again, this is very important for your UPSC. Ribosomes attached with mRNA to form a chain are called polyribosomes. Reserved materials in prokaryotic cells are present in cytoplasm as cell inclusion bodies which may contain phosphates, granules, glycogen granules. Gas vacuoles are found in blue-green algae and purple and green photosynthetic bacteria. And what about in eukaryotic? Of course, eukaryotic cells are present in protista, plants, animals and fungi. Cytoplasm is divided into compartments due to the presence of membrane-bound organelles. This is important, membrane-bound. The cell contains well-organized nucleus with a nuclear membrane. The genetic material is arranged in the form of chromosomes. Plant cells differ in having cell wall. So plant cell has got a cell wall. Plastids will be there. Large central vacuole as compared to animal cells. Animal cells have centrioles which are absent in plant cells. And now you got a plant cell as well as animal cell here. So what you need to do, draw the diagram, label it, and after having labeled it, now you understand the significance of each label. Like for example, you say nucleus. What is the significance of nucleus? Nucleolus. What is the significance? What is the significance of Golgi apparatus? Nuclear envelope. Plasma like this. And now, after drawing both the diagrams, plant cell and animal cells, labeling it, write the significance of each label, and I want you to post it tomorrow in our WhatsApp group. And I want very beautiful diagrams, not casual ones. Okay? And I'm expecting all the 100%, all of you will draw, label it, and post it in our